awesome tutor, and today we're going to be looking at cloning. Now, a clone is an individual produced asexually. Not sexual reproduction, asexual reproduction, and is genetically identical to the parent. Their genes, their chromosomes, their DNA exactly the same as the parent. And you should know that in cloning or asexual reproduction, there's only one parent, and that's how they are genetically identical. Because if you have two parents and there's sexual reproduction, you've got fertilization and so the mixing of genes, which makes the offspring not genetically identical to the parents. But if you have one parent, it's only going to be that one set of DNA, one set of chromosomes, so it's going to be genetically identical. We're going to look at plant cloning and animal cloning and the different methods of doing this. Okay, so cutting, that's the first method. It's self-explanatory. You cut the stem or a leaf from the original plant. It has to be a stem or a leaf. And then you give it the right conditions, and such as moist environment, until the roots start to develop. And of course, new roots and new shoots will form, and that is when you have your new, complete, small plant, which is genetically identical to the original. To make the roots develop, we usually use hormone rooting powder. And the main advantage of the cutting method as a whole is that it is very quick and very cheap. Now on to tissue culture, which is a more modern version. Tissue culture, the conditions have to be sterile. What does sterile mean? No microorganisms. Okay, we do not want microorganisms interfering with the process. We start out with a group of cells, a small group of cells from the plant. We add a specific hormone. You do not need to know what the hormone is. And a big mass of identical cells is produced. And after this big mass of identical cells has been produced, we put it in different conditions, the right conditions, and add a different mixture of hormones. And this stimulates each individual cell to form a tiny new plant. And so you can probably guess the main advantage of this is that we can produce large quantities of identical plants from just one single plant. However, the method is very expensive and of course quite difficult. Now we're going to move on to adult cloning, animal cloning. So animal cloning, first by embryo transplants. You need a fertilized egg. If you don't have one, get a fertilized egg. And then let the egg develop into an embryo. And then you divide the embryo into individual cells, usually with a glass rod. And then you let each of these individual cells develop into an embryo of their own. So you have different embryos. You start with one, and now you have more than one, because you divide them into individual cells, and then they form embryos of their own. For this to happen, of course, the original embryo, must be unspecialized. The cells must not have differentiated, otherwise the cloning process will not work. And then you place each individual embryo into the womb of a host mother. And of course, when the offspring are born, they are genetically identical. But genetically identical to who? Not to the parents, because it's by sexual reproduction, because you've got fertilization involved. The offspring are genetically identical to each other. So each of the offspring born, they're genetically identical to each other, not to the parents. And so, the disadvantage of this method is the characteristics are uncertain. Whereas, of course, with plant cloning, the characteristics, you know what the characteristics of the clones are going to be, because it's by asexual reproduction from one parent, one original plant. Now on to adult cell cloning. Okay, so for this example, we're going to be using aliens. Let's call this alien Bob and this alien Lucy. So we take an adult body cell from Bob, say a skin cell, and we remove the nucleus. But for Lucy, we take a mature egg or ovum that is unfertilized. And we remove the nucleus, but we don't keep it, we just want the cell body. We insert the nucleus of Bob's adult body cell into the mature egg of Lucy. The now empty egg. And now we give it a mild electric shock so that they fuse and it starts dividing into an embryo. And then we insert the embryo into the womb of a host mother, assuming these aliens have wombs, and the offspring that is born is going to be genetically identical to Bob. Now, a disadvantage of this method is it's very unreliable and difficult to go through the process, as well as it, of course, being quite expensive. That's it for cloning. Bye!